Okay, I see everyone. I see everyone still awake at this point on the last day. So I'm going to be talking to you about a pi calculus model of supercoiling in DNA circuits, because DNA circuits will supercoil, or they have supercoiling. It's an unavoidable part of DNA that currently is a little bit ignored in a lot of the modeling. So what is supercoiling in DNA? So DNA is made out of a double helix. It's twisted, and as it operates and performs actions, that twist changes and modifies. Almost all organisms possess negatively supercoiled DNA. That makes it a little bit looser, makes it a little bit easier for RNA to get at the base pairs, open it up and access the information for transcription. As you can see here, in its relaxed state, you get about 10.5 twists of the helix for every base pair. But when you transcribe the DNA, it's like you put a stick through the middle of it and pull it along. So on one end, it will get overwound and have positive supercoiling, and it'll make it very, very tight. On the other end, and it'll be completely loosened up and open, and that's your negative supercoiling. Right. So we get positive supercoiling ahead of transcription and negative supercoiling behind transcription. And this creates different regions of supercoiling within your DNA. There are various things that come along and try and fix that and relax it. And when you finish transcription and it lets go, it all sort of springs back to its original shape. But as you're transcribing the DNA, as you're reading the DNA, you get these different regions of supercoiling. And we can, in fact, generate these very purposefully. There is a lot of evidence that this particular gene, TET-A, will generate large amounts of negative supercoiling in a plasmid DNA. So in a loop of DNA, the TET-A gene will, because it's, and go easy on me here, I'm not the biologist, I'm trusting that they're telling me the truth on this. So the TET-A gene connects to a membrane as it's transcribing, and that holds that whole structure in place. And it means that the negative supercoils are even harder to get rid of, and they go even further through your plasmid. And it's a very big gene, so it's sort of always transcribing when it is transcribing. So if you have a TET-A gene in your plasmid, you get this huge negatively supercoiled domain in upstream of transcription. So where does this sit in synthetic biology? So currently we use a lot of gene regulation networks in synthetic biology, often to perform binary functions and OR gates, things like that. Oscillators and switches are very common as well. And this relies on two processes. So first, transcription on the DNA, which produces mRNA, which then goes to your ribosome, and you get translation, which produces proteins. Those proteins then interact back with the DNA and affect further transcription. Normally through repressors, so you produce a signal that stops other things from being produced. One of the problems we get a lot of the time with these systems is that once you put a few of them on the same system, well, suddenly they stop kind of working, or they work, but then they don't work, and sometimes they work, and sometimes they do odd things. And one of the reasons for this is that if you get too many positive supercoils, it locks up your helix, and nothing can get at that information anymore. So transcription stops. It stalls out. No more transcription for you. Whatever else your system was supposed to be doing, it's not going to be doing anything until secondary processes come along and relax it. But in the meantime, your whole system is stalled. The other thing that can happen is that your negative supercoils can get too high, which basically means that your DNA kind of unspools and destabilize, and then they kind of go running off and nothing seems, and at that point, it also gets in the way of transcription. So we kind of need to include this in our model if we really want to use these gene networks effectively and actually perform these binary functions. So when we were thinking about this and how do we include supercoiling, because one of the problems is that this transcription translation process, it's quite slow. You do this transcription, it goes off to the ribosome, and something comes back. And not only is it slow, it's an unreliable speed, because how long the ribosome takes to deal with that is however long the ribosome feels like, really. So what can we do with that compared to supercoiling, which moves, near instantaneously through your plasmid DNA? So we needed to model it separately, but we also needed to still be modeling everything else. So I looked at this and I went, well, it's sort of a signal. It's moving along the DNA and it's signaling things further along to change their coiling state. And so we've got biology. Everything in biology happens at once. They're not taking turns. And all of the genes are trying to do everything all at the same time. So we've got a concurrent system and we've got some signals. So if you've ever heard of pi calculus, it's a system for concurrent signaling. So they seem like a very good option in this case. And we can model all the things as signals. So we can model our transcription as our gene signaling out to our environment. And we can model our coiling as our genes signaling to an internal environment around the supercoiling. And we can have translation, which is the environment signaling back to our genes. Now, because I'm sure not everyone is familiar with pi calculus, you can see the raw maps of it in the paper or on request. I have some slides at the end that I can show you if anyone wants to see them. But it's mostly a state transition system. So you define all the possible states and they rely on some concurrent signaling to happen as well. And then you update the state of your system. So what are we going to do with this? Because now we've got this. Why don't we actually do something with it? Because now we've got these signals. Now we've got this supercoiling. Well, we could just use it to fix the existing things, or we could use it to make different things and do new things that might be a bit faster because, supercoiling is a lot faster than transcription and translation. And luckily, the biologists already had some ideas on that. So there we have here a design for a plasmid with a supercoiling sensor. So we've got in orange is the TET-A gene, over here. So that's going to generate our negative supercoils, and they're going to propagate backwards through the system. They're going to go past this gene initially, but then they're going to hit this guy. This is PLU500. He is a supercoiling sensitive promoter. So when the supercoils get to this point, it opens up, and suddenly M-raspberry happens. And if it gets a bit further, we hit another copy, and M-calama happens. M-raspberry and M-calama are supposed to be fluorescent proteins, so it should mean that the whole thing glows red or glows blue or glows red and blue. I checked, it's not magenta, it just glows red and blue at the same time. Again, not a biologist, don't know how that works. So if this works, that all works and it does what it's supposed to, but we want a circuit, so we don't want it to always do the same thing. So we've also got these lac operon points. Now, these are designed to bind with lac-I protein. And when they do that, they also bind with each other, and they make a nice little loop. So here we have the circuit without lac-I. And if that works out, the supercoiling propagates backwards, and raspberry turns on, and calama turns on, and we get this nice red-blue fluorescence. If lac is in the system, however, well, the supercoiling goes backwards, and raspberry turns on, but then lac jumps in here and creates a loop. And that locks that whole region up. And then the supercoiling can't transmit. So our blue fluorescent protein never turns on in the first place. And that's our nice, neat little test circuit that the biologists have suggested we start with. So that's very blocky. That's the design from the biologists. And we wanted to look at what does that look like computationally and with our pi calculus model. So we needed to sort of translate this a little, and we purposefully made it look quite different from the original, but a little bit like it, just to avoid confusion. So our gene TET-A becomes this gene TET-A, and this guy just produces supercoiling. He does nothing else in our model. In biology, he's also doing some other stuff, but we don't care about that stuff. So in our model, he just produces negative supercoiling. And we've got a fluorescent gene that's going to produce red. because we weren't too bothered about what kind of red it was, so red. And he couples, and it's coupled in with the promoter, so that's now one object. And we've got a blue one as well. And we've got this origin of replication because it's a plasmid, and apparently they have to have those. And it also apparently stops the supercoiling from looping all the way around the plasmid and doing really weird things. So that got included in the model as well, but it just sits there and stops supercoiling moving past it. And then we've got these two bridge points, which are our lac points. And we've got an anti-clockwise and a clockwise one of them. And as you see, they're connected to the middle by dashed lines. That's because these guys will sometimes stop supercoiling, but not always. Nice. So the full circuit looks like this. You'll notice there's a few things that weren't in the original circuit. So internally, we've got these supercoiling regions, which act as their own little objects. And their job is to get supercoiling signals from genes and to pass supercoiling signals onto genes and to each other. So S1, 2, and 3 are always trying to propagate supercoiling between them. So if S1 becomes negatively supercoiled, it wants to make S2 negatively supercoiled, unless something stops it. And our TET-A gene will be signaling to S1 to become negatively supercoiled, and S1 will be signaling the current state of supercoiling to our red gene promoter pair. But we also have our own little external environment set up because we don't have a ribosome. So we had to kind of invent one. And currently it's very basic, and we work on integers because we're waiting to sort out some stuff in terms of parameterizing this. And in the meantime, integers are as good as anything. We're hoping to get that a little bit better fairly soon because we've got a couple other models that are producing some results that we're going to use for that. So with LAC, this does what it says on the tin. And the negative supercoiling gets triggered by the gene TET-A. C red becomes negatively supercoiled, and our environment now has red in it. And if we move instead to our without LAC, then what? So just sit here for a little bit longer. You'll notice the bridges now have a prime on them. That's because they've changed state. So the bridges are by default open. Every single time step, they have to detect both the lac I in the environment and each other in order to remain closed. Because if you have one lac point, well, that's just been holding a bit of lac protein in, and it's not going to do anything. It's not going to form that loop. You need both of them there, and you need an ongoing supply of this lac protein in order for it to stay closed. So apparently it does take a while to reopen if you get rid of the lac protein. Apparently that's a surprisingly strong bond. Right. But if we don't have that, if there's no lac in the environment, then the supercoiling will propagate right the way around down to our third region, where it doesn't really do much, but in the second region it turns on blue. So now we have some red and some blue in the environment. And after a while, the difference between the amounts will become negligible. Yeah, and that's our neat little circuit with pi calculus for now. So we've got the beginnings of this pi calculus representation. We've got this neat new visualization that does not confuse biologists into thinking it's actual biology, but also is quite readable by the biologists, which was very pleasing. They like circles, it turns out. Well, circles to represent circular things. And this gives us a computational description of what's happening in this plasmid that's nicely abstract from the biology, and we're hoping will help us to identify different components and how we can use the supercoiling as a signaling mechanism rather than as something that just interferes with all our systems. There's an implementation of this existing system available on GitHub, and we are actually building these. So we have collaborators in Ireland, Aalap, who is building all these little plasmids and putting them into E. coli and Salmonella. And and I'd love to have results for you on that, but we are currently in the middle of literal debugging. So we've literally taken the plasmid out of the E. coli and Salmonella and sent them off to be looked at under a really, really cool microscope thing. So this is pictures of our actual plasmid as seen in really cool magnification. And that's to work out why they're currently really not behaving. Aalap's got a lot of ideas on why that is, and he's producing a lot of variants to work out what's correct. And we're hopeful that we'll have a solution for whatever's going wrong there fairly soon. And that is and so he's building what we call our biological model, because while it is reality and it's actual plasma is actually doing that, what we see of it is functionally a model, because there's only so much we can actually manipulate it, and there's only so much we can read out of it. So for instance, those plasmids, all the readouts are done at a colony level. So we only see these statistics over the top of them when we're looking at that. We've also got a physics model. Now, that's Victor's stuff. Victor's doing a great job with that, but that's at the molecular level. So he has literally got a model that twists and untwists the DNA. It literally has individual things hooking onto that DNA and transcribing it, and all sorts happening there. It's a really cool model. I'm hoping to use it to parameterize this model just as soon as we can work out how to get everything properly compared with one another. And that's the other thing we're doing, is we're looking into how do we formalize doing comparison between these three models, because they're obviously very different from one another. So how do we parameterize between them, given what we can actually read of each of them? So that's going to bring about some probabilistic state changes, hopefully, on this PyCalculus system, and real numbers for our transcriptions, so we can start to deal with the timing differences between transcription and translation, and supercoiling, because supercoiling is near instantaneous and transcription can take hours, and that's before we even get into the translation problem. So yeah, and hopefully once we've got all of this going a little bit smoother, we're hoping to add more components and do some more complicated things, because we think we might be able to put a second bridge setup in and do some cool squidging of our plasmids into weird shapes to do weird computation. So quick thanks to the entire talk team, all 7 of us, as seen in these pictures, and the PSRC and the Science Foundation Ireland for funding all of this craziness and letting us play with some fun bugs and their DNA. Thanks.